Our lab studies evolution using these stickleback fishes as model organisms. They protect themselves with erectable spines. For many traits, we have identified genetic changes underlying their evolution. For such studies, measuring various lengths of a fish is a routine. However, to attain statistical power, hundreds or even thousands of fish must be measured, causing psychological damage to vulnerable grad students. For their well-being, I built FishPose, a model that identifies the XY coordinates of these key anatomical marks given a fish X-ray image. FishPose was adapted from DeepPose, a human pose estimator. Compared to humans, fish postures are much less flexible. Furthermore, the input images are also clearer due to their scientific nature. On the flip side, the dataset size is limited. Therefore, the goal is to build a simple model that deals with rather standardized inputs with limited training data. The final model is a 6-layer convolutional network. Preprocessed inputs pass through 4 convolutional layers and 1 dense layer, with the final layer outputting XY coordinates of key marks of the fish. Trained with 3,000 fishes, the model performs reasonably well. Here, the dots denote the ground truth, with lines extending to the predictions. Although there are cases where the tip or base of a spine was wrongly predicted, it looks okay overall. In this plot, we can see that individual predictions often fall within a few pixels from the ground truth. Not bad. However, many skeletal elements are short, and a few pixels deviation can result in over 20% error in length measurement. This prompted us to ask, what can such a pathetic model do? To illustrate that, let's look at this graph, which shows the genomic regions responsible for length variations of these 2 spines. This analysis requires the length measurements of spines from a large number of fish. We can see that the same analysis using measurements produced by FishPose performed equally well. Remarkably, it identified not only the major peak on chromosome 4, but also secondary peaks on chromosomes 13 and 6. This demonstrates that, with its mediocre precision, FishPose is qualified for such genome-wide scale analysis. The FishPose was trained on sticklebacks. We hope that, with a little tweak, the model can be applied to other related fish species, or even animals with legs, as long as they are of interest. By doing so, we hopefully can save grad students from a broader research community.